I'm really happy to follow Derek's talk. I feel he and I are the same vintage, I think, an experimentalist. We feel like Waldorf and Statler, you know, the two grumpy old men in the Muppets. I hate your stuff. So we have a sort of very interesting stage and almost in human history, right, after the genome was done. And it sort of confronted scientists, biologists like me, to reveal that the world is finite. There are 20,000 genes. In a million years from now, there'll be 20,010 or something like that. But we haven't really changed the way we do biology experiments to confront the reality that there is a box in which we work. And so when you're talking about failing in phase 2 trials, it's the only real ground truth test of how much we understand human biology, because the only time we intervene with a hypothesis and get an answer. The stuff we do in the lab is a lot of fantasy worlds, right, of how the cells work and how the cells don't work, and we construct a reality that we can play with. And if you think about, can we really model biology? Well, I always go back to the point that 10,000 genes in us have fewer papers and fingers on my hand. And how in the world can one construct a model where you absolutely know half of the information is missing? It makes no sense to me. And then into this world comes AI. We don't need the data, we don't need clean data, dirty data. We can solve everything. And it makes me vomit. And so, you know, as a structural biologist who contributed, we contributed about 10% of the data to the PDB, the organizations that we've been part of, and we saw that rigorous effort that did have a success in being able to make predictions. And I can tell you, it's $10 billion of really hard experimental work that created the instant success of AlphaFold. It was 27 years of John Molt comparing progress every 2 years, very dedicated. And that's the kind of science that leads to these instant advances. And what I fear today is that we're trying to leapfrog past the hard work and go right to the, don't worry about the data, just give us more data, we can handle it, we can find the noise, don't worry about us, which is obviously not true. And so how do you sort the sort of public's perception, even scientists' perception, biologists' perception of how you can go from these messy data to an AI-directed solution? It's very hard to explain it to them. You say, you know, those RNA-seq experiments are too noisy to find patterns. No, you cannot predict what a microglia is because you don't know what a there is no ground truth. It's very difficult. So in that world of complexity, you have organizations that are rigorously comparing progress to define goals, setting up the questions correctly, monitoring the progress correctly. And that's what we're going to talk about today. Organizations like the Dream Challenges, which Gustavo led and started, for 20 years have been thinking carefully how you statistically and carefully and transparently measure progress, define a problem well, and then get us to the end. And I think we're going to need more and more of those organizations who put a sort of a sense of reality to what is going on out there. As I said, to me, it just is illogical. We can model biology, we have half the genes we have no data on, and there is no ground truth. How in the world can you build a model? You can see anything you want. As Derek said, you see patterns, and we're really good at that, right? And so today, I'm going to introduce Gustavo, who was at IBM for a while, is at NYU now, and who's been thinking about this for ages. And I think we're going to get some really good insight into how that field of critical assessment, shall I say, evolved over the last years, because he's an older guy now.
Hello. Okay, so I would like to talk about how to think about rigor in predictions when we do predictions from either AI or from other basic principles. So that's what my talk is going to be about. So the outline is going to be, let's talk a little bit about the crisis of reproducibility or replicability, of however you want to call it. And then how we can benchmark or do critical assessment, as has been called, methods that purport to solve a predictive problem how do we benchmark those and understand the limits of reproducibility of those? What additional problems exist in the evaluation of predictive models, and a proposal for integrating the many benchmarking efforts that have proliferating for good reason, but now there are too many to control. So I think Derek was talking about this before, right? AI cures all is the new oil snake. Snake oil, sorry. So let's talk about this paper that was published in 2012 that was really an eye-opener for many people. I don't know whether you remember this paper or you have it in your radar, but the idea was these guys, I think they were from Amgen, took 53 papers on preclinical oncology and said, they were relevant for their drug discovery purposes, and they tried to reproduce them in their labs. And of those, only 11% replicated, and the rest didn't. And that was a big surprise. How can it be? And it's not that they didn't try. They tried hard. They brought the authors of the original papers to try to see, what are we doing wrong? And they couldn't replicate. And this is not the only situation. This has been addressed since that paper and onwards. Last year there was this paper on models that predicted clinical trial outcomes within clinical trials, and then they got, a balance accuracy of 70% or so. But then when they used that clinical trial and tried other clinical trials, they got pretty much random level prediction. This is another one that was interesting. EPIC, which is a health informatics system to keep all the records, electronic health records of patients in different health systems, is probably the dominant. EPIC is the company. They created a sepsis model, which predicts within 6 hours whether a person is going to start to have all kinds of organ failure. And they got a reasonable area under the curve of 76 to 83%. That was the confidence interval. And it was used. But then sometime later, someone said, let's try it in other university, not at the University of Colorado, at the University of Michigan. And they got 0.63 area under the curve, which is pretty bad. If you are a sepsis patient, you don't want that area under the curve. And by the way, one of the reasons why this happens is because the health system processes change. And so the test data is different from the training data, so things evolve. We will discuss this a little bit later. More recently, there have been some interesting papers trying to discuss what are the problems, the leakage problems, the p-hacking problems, there's so many specific details of how you train your models that may lead to this lack of reproducibility. But this is a thing, right, that we have to contend with. Well, we can look at this, like I was thinking, the glass is half full or half empty, right? So, but the reality is that there are reasons why to think that the glass is half empty. So a while ago, like Al mentioned before, we were thinking about these kinds of things when we were looking at systems biology, which is the field where I belong, and we wanted to say, there were a lot of people trying to predict things, gene regulatory networks, protein-protein interaction networks, and think of the 12,000 genes that we know, not the 8,000 that I'll mention, as a system. And so we said, the problem was that, for example, suppose that I do a gene regulatory network inference. And what used to happen, and sometimes happens today, is that, okay, I have, 1,000 genes, and I have 10,000 possible interactions between this gene or gene regulations. And then I will cherry pick a few and I will say I validate because I go and find these 10 cases. I mean, I plead guilty to that as well, right? That is how, we started to think, I want to see whether some of these are good. So I go in the list, I choose the ones that match some of the existing literature, and I cherry picked, but I can publish. And that has happened. So I said, this is not the right way to do science. You have to, for example, if you want to validate 100, you have to pick 100 at random or with some statistical criterion. So we created this DREAM challenge. DREAM stands for Dialogue for Reverse Engineering Assessment and Methods. Dialogue is because we believe in open science. Reverse engineering is because we want to know it's not just predicting, but understand what's going on there. But Derek, one question I have for you is, what do we mean by understand? But that's something else. So the DREAM challenges, then, this is tries to be a community. Again, a word that we need to define more carefully. What do we mean by a community? But we mean a community that thinks together about how to solve problems. And these are the missions of the DREAM challenges, is to contribute to the solution of important biomedical problems. That's one of the things that also Derek mentioned in his talk is, what problem should I be working on, right? What questions should I ask out of the data? And which data set should I collect that addresses these questions? And also to objectively assess algorithm performance, because we do blind evaluations that provide some unbiased method assessment, but there are problems with that we will discuss in a second. And then make an apples to apples comparison to help discover the best methods. Not because we will only use the best method, as some people think, but we want to know the limitations of different methods and compare them between them. And provide evidence about the solvability of a scientific question. Sometimes we cannot, we have run challenges in which nobody did better than random. And why is that? Is because the methods are bad, or maybe we are using the wrong data for that question, right? So though it doesn't show the impossibility of solving that problem with the data at hand, it does indicate that we need to think harder. Fundamentally, we want to do competition so that people collaborate, which is kind of a contradiction in terms, right? But usually we try to foster scientific collaboration by making people compete, and once they say, I won, I'm the alpha dominant person here, then you start to feel a little better about collaborating, and we do a collaborative phase where we do our paper writing, our thinking, and so on. Democratization of data and acceleration of research are byproducts. I think there is no need to put this slide, but just in case I do, because some people say, yeah, what do you mean by benchmarking, ground truth, and so on? Some people ask me when I say, we use a ground truth to benchmark, and say, but why do you want to make a model to predict something that you already know, right? And I always found that surprising that some people ask me that. But typically, we take a biomedical data problem, typically labeled, and we know the outcomes in that one in one or more choices. So if this is an organization, it could be DREAM, Cash, or even a pharmaceutical company. Like we did challenges with different pharmaceutical companies with their data in a model to data kind of paradigm, so that their data is not leaked. And we crowdsourced to as many people as possible. This is the community that we want to reach. And they are, typically the question is, what are the incentives for these people to participate? And we can discuss that at some other point, but they want to know also how accurate their algorithms may be to solve the problem that they gave them. And they propose solutions that we compare with our ground truth. And that's the way in which we try to determine which one is working well and the degree to which they are working well. And we have written several perspective papers on this. And we have done many challenges, and typically all these challenges have a collaborative phase in which we do paper writing and try to justify, you know, and try to find a little bit more of, it's not just a horse race, you know, John won. It's more like, what did we learn from it, right? And so typically we manage to choose problems and find algorithms that are sufficiently good to publish in good journals that will motivate the next generation challenges to say, okay, if I participate in dream challenges, maybe I can publish. And that is one of the incentives, the carrot. But I'm not happy with just publishing, right? What is the impact of the things that we are doing? And this is what we are trying to emphasize going forward. It was mentioned this. So Jeff Hinton said society should stop training radiologists. Radiologists would be replaced by software in the next 5 years. He said that in 2016. And I work in a health system, and radiologists are happily still looking at X-rays and MRIs. I wanted to show, it's not that it's not possible, but it's like we are not yet there. So I wanted to show you a challenge as an example to show how we used a benchmarking competition in breast cancer screening. And the competition was basically, this is the paper that resulted. It was try to determine the cancer status of a woman given a screening exam, a panel of clinical and demographic information, and if available, previous screening exams. You get all that, and you want to know whether these algorithms, using all this data as training, will be able to predict a hidden data set about the ground truth that this woman had or didn't have cancer in the next year. So that was the label, we knew that. And so what we ask is that everybody give us your best guess or belief that the left breast or the right breast had a lesion, a tumorous lesion. And by the way, we offered $1 million to those who won, not we offered, but the Arnold Foundation thought it was a good idea to incentivize this, but only if the best performing team did better than the radiologists on this same data set. And the data set came from Kaiser Permanente and also from the Karolinska Institute, another test set. We had of the order of 90,000 women and 150,000 exams, because some women had more than one exam in time, of which 1,000 were cancers, and we had more than 500,000 images. So it was sizable data set for the time. And here are the results. In the y-axis, we have the specificity of the algorithms at the radiology sensitivity, which, if I remember correctly, was something like 81, 82%. This was the specificity of the radiologist, about 90%. And this was the specificity versus the area under the curve, by the way, of all the algorithms that we evaluated. So nobody got to be to have the specificity of the radiologist. So nobody got $1 million, but in the attempts towards that $1 million, some people got 100,000 or something like that. So that was, we were happy to give that money to those participants. The other important thing is that thing about replicability, right? We separated a chunk of data from the Kaiser Permanente, but also had a chunk of data from Karolinska Institute, from a collaborator from them. And we wanted to see whether the performance of these algorithms, each of these points is an algorithm, compared between Kaiser Permanente and the Karolinska Institute data set was comparable. And And what we can see here is that they were pretty comparable, even better in the data set that they didn't see, for reasons that we try to understand. But this is what we need to do, is to, of course, test the data set in the same training, in the same cohort that the training data set was taken, but that was not, that I don't think ever is enough. You have to test in other data sets to not fool yourself that there were bias in your original data set. And this is an example of how it can be done. But again, I don't think that 2 is enough. It has to be many more, but for the purposes. Now, the glass was half empty because we couldn't get to the same performance as the radiologist. But we believe in what is called ensemble learning, or put together algorithms to compensate each other's deficiencies and make the aggregate be better than the best of the algorithms that you are aggregating. This is what, in some other contexts, is called the wisdom of the crowds. And so the area under the curve in the validation set of the best individual performer was 0.88. Of the ensemble that we did was 0.89. And for the radiologist was 0.94. So they were much better than the algorithms were. But then what we did was we put together, we ensembled the radiologist assessment with the algorithms assessment, with the ensemble of algorithms assessment, in order to determine the degree of the belief of the aggregate that the woman that we will look at had or not cancer. And what we got is that we went up from the specificity of 9.905 for the radiologist alone to 0.92 of the ensemble of algorithms and the radiologists. It's only a 1.5% increase. But the negative set of a screening is like 40 million. So 1.5% in 40 million is a lot. So this is important when you are looking into screening or diagnostics. And that would mean like 500,000 people not being told you have to come back because you may have cancer, or zero. That's what we are trying to do. That means this 1.5 may seem little, but because of the nature of the problem, then the negatives are much bigger. And so an increase in specificity means a decrease in false positives, which is very important. Also, it has economic value. Okay, so this was an example of one of the almost 100 dream challenges that we have done. These are the challenges that are coming. The Dream Target 2035 drug discovery challenges that we are doing with data that Al is and his team is producing. Please go to dreamchallenges.org to register to in this challenge. We have a Dream Olfactory Mixture Prediction Challenge, and we have a causal diagram. I mean, trying to start to think beyond just AI and start to think more in a causal way, how the treatment of COVID depends on different characteristics that the patients had. But if you are interested in any of these, please go to dreamchallenges.org. Now, I told you about what Dream is doing or has done. I'm telling you what we are not doing and the problems that we should try to solve together. So one of the things that we are not doing is that algorithms have what is called temporal degradation of performance. The sepsis model, for example, was tested for some time, and when it was tested downstream, like 5 or 6 years after it was trained, the performance degraded considerably. And the reason was that the ICD codes, for those of you who know the diagnostic codes, had changed from ICD-9 to ICD-10, unbeknownst to the people using this algorithm. That means that the training set was no longer valuable. But the same thing I'll show in a second, happens with a lot of algorithms. It's not that time is the parameter, it's that the training set becomes more and more deviant with respect to the test set. And therefore, we need to be aware of that. We are only maybe testing in one holdout data set or in two, but we have to do it across many data sets. We have to do a federated tested approach. And then is the problem of evaluation of foundation models, which is a disaster because we don't know how to benchmark foundation models because they purport to do so many things. So how do you validate those so many things? This is the temporal quality degradation. So in a paper that was published a couple of years ago, you train at some time and you wait some time and you test. And you do that for different machine learning models, for different predictive models. And this is what happened in this paper. This is the error, the RMSE at the initial time. That means when this future is really very close to the training. But if you take the average or the 75 and 25th percentile of the RMSE, you see that the error grows like 80% for some algorithms. For some algorithms, a little less, but it changes, it degrades. Furthermore, it depends very much on the model that you are using. For example, this is for a weather data set that is predicting the next day temperature and humidity. For a neural network, it went from 1, which should be at the beginning, to 100. I mean, why is it degrading, right? It trained well. It had a good performance at the beginning. Anyway, I'm not going to say why it was degrading, but I want to say machine learning algorithms trained today might not perform the same tomorrow or in 5 years. And this is very important when you use it in healthcare. The other thing is that we need to use a federated network for benchmarking, as some efforts have been doing, because the fact that it works well in your data set or in your holdout doesn't mean that it will work well in many of the others. And we need to understand the specificity of the location of where the data was collected in order to see whether the algorithms generalize. And then we come to foundation models. This is an interesting paper published 4 years ago, about opportunities and risk of foundation models. And this is a long paper. You can summarize it with ChatGPT and learn it. But let's discuss some of the things that they discuss, like, for example, the philosophy of understanding. But before going there, these are a lot of the by the way, this is AI-generated list of foundation models, or structure prediction, protein and peptide binding, what else? Cell models, perturbations, target discovery, all kinds of things. And if you think of these foundation models, what they do, and I'm going to kind of shift a little bit gears and think a little bit of the history of science and how astronomers used to think of the orbit of planets in the sky. The early way of thinking was using epicycles. The Earth was in the center in our geocentric worldview, universe view, and then there were a deferent, which was this big circle, and this planet was turning around this center in the deferent, which is a completely correct model if you think that from our perspective, the sun goes around us and the planets go around the sun. So it's not that the model is wrong. The problem is that it was a fit, but also it was wrong in the sense that we have elliptical orbits, and so this was not enough. So what happened is, for example, when they started to look carefully and they saw that the retrograde motion of Mars, for example, was 83 days every 786 days for the orbit of Mars, they said we need to change this a little bit. And so, this is the Ptolemaic model. He started to say, okay, let's add to the epicycle another epicycle, right? And this corrected a little bit the timing of that. But there were other problems that surged. And so eventually the number of epicycles for the 5 planets that were known in the time of Ptolemy went to 80. And this is the universe according to Ptolemy. It was a little mess of epicycles and turns around epicycles. But this wouldn't be bad if you predict. So you will say, oh, this is good. Now this is the analogy that I want to do. In the early 1800s, Uranus was observed to have an orbit that didn't move as expected. It was a little slower and then a little faster. And so what happened was Newton had already proposed the theory of gravitation. So this French guy, Leverrier, said maybe there is another object out there that is pulling gravitationally on Uranus, and then that would explain. And so he did the calculations, and he said to the astronomers, you had to look there. And when they went and looked there, they found a new planet, which is now called Neptune, 1 degree different from what Leverrier was proposing. But this is a success for the theory of gravitation. But why am I telling you this? Because if we were in the age of machine learning, what we would have done with this aberrant observation of Uranus is add another epicycle. And this is what machine learning does, right? When something doesn't work, well, let's put more data that includes that aberrant thing, and now we are fitting it. But we are not understanding the laws of gravity. We are just patching over patches and over patches. And that's the problem that we are having with using machine learning. Now, sometimes we need to think, if you want to validate, like we are in the business of validation in doing dream challenges and other things, we need to think about what do we mean? How do we know that the model is right or not? And so you have to inevitably go to the philosophy of science and to epistemology, and to think that there are differences between a theory that purports to be a scientific theory and machine learning or a statistical truth. In principle, there is predictive power versus explanatory depth. So AI focus is on practical outcomes, predictions, without needing to explain mechanisms. Science is explanatory, seeking to predict and to understand the principles underlying natural phenomena, so that when you see something that you didn't see, you can explain based on those basic principles. Data-driven learning versus theory learning understanding is that AI follows a data-driven approach, as if everything that we need to know is in the data we have, except that we don't have data on 8,000 genes, and we don't know what we don't know, without needing to understand about underlying system. Science is theory-driven, where models are built from general principles, deductively, or hypothesized from particular observations, inductively, and then tested against empirical data. And then you have the Popperian way of thinking, which is, okay, models or theories can be refuted, cannot be validated, really, because you need an infinite number of experiments to validate. So performance validation versus falsifiability, AI lacks the emphasis on falsifiability. We cannot falsify an AI. You know why not? Because if I say, oh, this diagnosis is wrong, and then, well, it's a false positive, or it's a false negative. And then you change the false positive or sensitivity or specificity, but you are not learning anything about the underlying causes why this is not working, right? So that's why we need to think about what does it mean to verify or to validate an AI model? So AI lacks the emphasis on falsifiability, which was Popper's scientific method. AI models are often judged based on their empirical success. And we come to George Box's dictum that all models are wrong, some are useful. And you can go and read in literature, these, for example, single cell foundation models are they really suck. They are not good, really we have problems, and we don't know exactly how to validate. So we can benchmark them, we can see the limits, but we don't understand its really limitations. So the question is, do we need new critical assessment efforts? And clearly, there is a brave new world that is coming with new advances in genomics, proteomics, cryo-EM, CRISPR-I, DNA encoding libraries, and so on and so forth. So we have a lot of data that we will chew. But these innovations need new approaches to ensure clarity and rigor that we don't exactly understand what those new approaches are. Now, critical assessments, as I have shown at least in some examples in the DREAM challenges, are methodologies that emerge as a scientific methodology to contain our ability to understand the limits, right? And have already demonstrated enormous value, as we know very well from the CASP example. So we have had a first generation successes in the 30 years of CASP, in the 20 years of DREAM, and many other. And so our proposal for the next steps is to integrate and build on this foundation. Integrate, not make DREAM bigger or make CASP bigger. Make us bigger by joining efforts between CASP, DREAM, Polaris, whichever methods we can put together if it makes sense. So this is an example of the Chen-Zuckerberg virtual cell model in which we have different descriptions at the DNA, RNA level, protein level, then embedding that allows a representation of those levels, then go to a phenotypic readouts that allows another representation, and then you have to think all this is intracellular or macroscopic, but we need to think of spatial interactions and cell-cell interactions and so on and so forth. This is a beautiful concept. But the problem is, how do we know that it's good, it is correct, right? And so, well, today what we have is that, for example, the critical assessment for genome interpretation or CASP or functional annotations or DREAM, they are all doing something, but they are not looking at the whole picture. They are only looking at, like in Derek's analogy, the little skirt of this lady, not looking at the bigger picture, right? We need to try to think of how to, if we are going to create a virtual cell, how do we validate it in all its glories, not between go from, you know, the transcriptome to the viability under some drug. No, that's kind of jumping too many layers. The thing is, how do we validate that? And so we believe that integration of critical assessment efforts really could help, have many benefits. For example, we would consolidate this challenge ecosystem with possibly a one-stop shop for benchmarking. Not, I insist, because I don't want to sound like, oh, unless let it be DREAM. I don't say that, right? Let it be whatever results to be the best, but we need to work together, like we are trying to do here with open science. Maximize opportunities for synergy, enhance industry engagement, make these efforts sustainable. We are always thinking, where are we going to get funding in order to do the next challenge? And we are always concerned about our sustainability. Increase the clarity in the critical assessment ecosystem in terms of making it easier for experimental communities to give their data to one bucket, and within that bucket, we would determine what is the best way to validate and what to do with that data. And one of the interesting things I think, so we can, of course, consolidate all these efforts in a coalition and do different tasks like drug discovery, disease mechanism, precision medicine, as we have been doing. This is more of the same. But more importantly, create a think tank to try to think what is the best way to advance the science of benchmarking. Think about that. It's not obvious. It's not more benchmarking. We need to think out of the box. For example, these are examples of people who are thinking how to do the benchmarking of these things. So this is a recent article in Patterns called Leakage and the Reproducibility Crisis in Machine Learning-Based Science, and they surveyed a lot of papers that use machine learning, and they classified errors into 8 different types. They made an effort in reducing the entropy of all the possible problems and proposed models to, like this is very annoying for us who sometimes publish in these journals where they say, tell me how you did this analysis and how did you measure this and how did you do this and that. It's very annoying, but it's so necessary in order for things to be reproducible. So we need to do the same thing for AI. And this is even more interesting to me is a perspective which appeared in JAMIA on how AI can be thought of as an intervention in the health systems and thought of it in a causal way. An intervention is when you modify the system and you see the results. Well, using AI is that. And so can we understand the implications of using AI in the context, workflows, in this case of healthcare system? So to finalize then a few words of summary and conclusions. So critical assessment efforts have created sound best practices to rigorously evaluate predictive models in medicine. But evaluation is not validation. More needs to be done to ensure generalizability in time, in location, and in the blackness of the foundation model box. We need to understand more about that. And we believe that a coalition of critical assessment efforts put our heads together to think how to solve this problem of validation can consolidate the challenge ecosystem, maximizing synergies and sustainable funding. So thank you, the Dream Team, John Moult, Al Edwards, and Luca Foschini, with whom we have been thinking about this coalition, and others as well. Thank you.
The next speaker is from Montreal, hooray, my hometown, and this is about a competition and a sort of a structure that wants to work on drug discovery problems, together with industry, which I think is super important because there's problems that are academically interesting and problems that have real world interests. And I think that, if I'm correct, that the idea of the organization that he's championing, Polaris, is to work with industry and define real world problems. And he comes from now an organization, or now a larger company, where I met the CEO and he said a really interesting thing to me that I really like. In all the experiments previously, data were the exhaust of the experiment that were thrown away to get to the result. And the way that they're thinking about it now is to collect all the data to be able to make better predictions, which I think is a really healthy way of doing it. And so today we're going to hear about, I guess, Polaris, which is a super cool initiative, close to my heart because it's the area of science that I do. So welcome, Cas.
All right, good morning, everyone. I'm very excited to be presenting Polaris today, and as it was already hinted towards, Polaris is this benchmarking platform. If you're coming from machine learning, you may be familiar with CASP, you may be familiar with Kaggle or Hugging Face. It's a good mental model, but we've built it from scratch specifically to support the needs of the drug discovery community. Before we dive in, though, I wanted to ask a more broad question and take a bit of a step back and to ask all of us here, like, what actually makes for a good benchmark? And so one success story that comes to mind for many of us is CASP. CASP has been this competition that's been running for a while now. It's every 2 years. And I think it's been recognized by many as a driving force of innovation by consistently identifying valuable innovations within our community. It's gotten a lot of attention because AlphaFold demonstrated their model and their results on this competition. But I would actually argue that its impact has been underappreciated, has been understated. And a good example of that is this very plot. This is a plot that was used to communicate the results. But forget about all the data that's actually in the plot. I think something that we've taken for granted is the axes of this plot is something that we've known for a long time, but it's actually not the case, right? Like, I think that one of the main contributions of CASP is not only that they've identified valuable innovations, but they've actually improved upon the methodology of benchmarking a lot, right? So if we look at these axes, the y-axis is performance. That's something we've refined through, or I say we, I played no role in this, but like it's something as a community, we've refined throughout many years of running this competition. And similarly, the x-axis, like the difficulty of the data point that we're trying to predict, is also something that we didn't quite know how to do. And it really took a group of dedicated experts to rigorously analyze these results and to figure out, like, how can we come up with a way of benchmarking methods that just better reflects the problem we're trying to solve here? How can we pose this question in an even better way? And they didn't stop there, right? Like, all these performance metrics, these aren't trivial to implement. There's a lot of alignment that needs to happen in pre-processing. There's a lot of edge cases that need to be addressed. And so it's not trivial to implement these metrics at all. But what they did is the organizing committee, they went above and beyond, and they provided the community with open source software to implement these methods in your day-to-day science. And even more so, there's various community initiatives. So they didn't assume, like, if we're doing good science, people will pick up on it and like the word will spread automatically. But they actually made a proactive effort to try disseminate the results of these competitions within the broader community. And they did that through special issues of journals. They did that through conferences that they organized. So they put in a lot of effort to not only provide substance, but also form, to make it easy to distill these results and to understand the outcomes of these results. And I would argue that that has really matured the field of protein structure prediction. This has laid a foundation that a lot of others can build upon. And there's here, I'm highlighting 2 recent papers. I think one was mentioned in the Q&A of Derek's talk, which is runs and poses about, like, measuring if protein ligand models, if they're actually, like, generalizing or memorizing, like, the metrics, the similarity metrics, the performance metrics that are used are based in large on, like, what CASP has developed over the years. And I think this is indicative of a broader, like, pattern or strategy. I think if you want to be a successful, if you want to propose a successful benchmark, you need to address these 3 pillars. So you need to figure out, like, what are the best practices. That's likely something you can't do all by yourself. This needs to be a cross-industry or, like, at least an open science collaboration on figuring out, like, what to do, how to benchmark these methods. And you're probably not going to get it right on the first try, right? So this needs to be open for community feedback. It needs to be something that can be refined. But you can't stop there. You can't just publish a paper, even in some high-impact journal, and call it a day and expect that everyone will run with it. I really think you need to go beyond publishing papers. So what can you do? Well, you can provide tooling. So if we think about the guidelines as the what, the tooling is really the how. How do you make it easy for people and lower the barrier of entry for people to adopt these best practices in their day-to-day research? And then the last thing is community. I'm not entirely happy yet with that label, but we evolve our thoughts as we go on. But, like, what I'm trying to say with that is that we also need to provide people a reason to implement these best practices. It creates friction. We're going to ask people to change the way how they work. It may not be easy. It may be extra effort. It may be more difficult to get published if you do these things, right? Like, so we need to figure out ways to create incentives for people to want to adopt these best practices in their research. And those 3 pillars are core to our strategy with Polaris as well. And so to address the first, guidelines, we have formed what we are calling steering committees. So first of all, what's Polaris? Polaris is a benchmarking platform to assess machine learning, specifically in drug discovery. It's an industry-led initiative. And so the steering committees are really these cross-industry, interdisciplinary groups of people that come together to establish best practices based on the problems they're facing in a real-world context. And so, as I was saying, that this hints towards this like first pillar, guidelines. It's really about like we're trying to figure out how to do benchmarking. What are these best practices? And we've had some initial success. So our scope, we didn't start out wanting to do competitions. We figured competitions are, well, we see the value of competitions, right? Like you're disentangling the method evaluation from the method development. There's this blind test set. It's just a more unbiased way of doing things, and that's great. But they're also really expensive and resource intensive to run. And what that is leading to is the reality is a lot of people just work with these retrospective benchmarks. So we were thinking, can we come up with a way to extract more value from these retrospective benchmarks to make those more impactful? So that's our initial scope. And also we didn't want to solve all of drug discovery. That seemed like too big a task. So we explicitly limited the scope to small molecule property prediction. And so we brought together a group of people and we identified 3 topics that we thought we could make valuable guidelines, valuable best practices, and that's method comparison, measuring generalization or data splitting, and data curation. And so the group of people we brought together are really cross-industry. And I can quite proudly say that all of these people have been proactively engaging with us, right? If I go on holiday as the one that started this initiative, I sort of expected it to fall flat for people to stop working and then I would get it. But actually when I went on holiday for 3 weeks, I got an email asking how we can please make sure that we keep up momentum. And the reason that all these industry partners are so excited is because they face this problem in their day-to-day lives, right? We had this discussion on like 11% of the papers are only valuable. Like that's something that's true in industry as well. Like these teams have spent significant resources trying to reproduce results only to find that it fails time and time again. So like they're excited to find a way, even if it's not perfect, to get more value from the work that's happening in the scientific communities. And so what do those guidelines look like? Well, we published our first preprint in fall of last year. And I don't want to go through it in detail, but like this was all about method comparison, how to do cross-validation, how to do statistical testing, what performance metrics to use. And it's really meant to be very easy to read. So like I'm including one of the figures here, which is really designed as a graphical table of contents. So it's this flow chart. You can go through it, figure out what applies to your experiment, and hopefully it's made really easy to then adopt these best practices in your research. And as I was saying, we're going beyond. We have code examples, and we want to make this easier, as we'll talk about in a little bit. Now, one thing I want to already clarify is like, we call this an industry-led initiative. I think that's one of our unique angles, but it's really meant to be community-driven. So when we published this preprint on method comparison guidelines, we didn't just want to like sit in our ivory towers and tell the people how to do their work, like not at all. We realized that we didn't have all the answers either. So what we did is when we published the preprint, we had this open feedback period where anyone could come to GitHub and just share their thoughts on the work that we had done and then challenge us and critique us. And it's been lovely, honestly. Like we've had a lot of good discussions, a lot of people that had a lot of good insights. So I really feel like that open science process has made for a better set of guidelines that we're now working to incorporate in the paper and submit it for publication. This is a bit of a sneak peek. We've had quite good success with the small molecule steering committee. Like I was saying, I'm quite proud that everyone is very proactive, and we've seen some first results. We've developed a bit of a playbook of how these collaborations could look like. And I'm excited to say that we're broadening our scope. We're also trying to find out, we're also forming a steering committee in the space of multiomics. More on that is coming soon. Okay, so we've talked about best practices, guidelines, like how to do your benchmarking studies. Then the second pillar is tooling, how to make this as easy as possible. And this is where the Polaris Hub comes in. So the Polaris Hub is a website. It's a benchmarking platform. Like I was saying, similar to Kaggle or Hugging Face, but it's built from scratch to make it really easy to do the right thing and really difficult to do the wrong thing. It has 3 primary artifacts. So we have datasets, benchmarks, and competitions. So a dataset for us, you can think of it as a table. So we have the rows that are individual data points or samples, and then the columns are different variables or measurements for those samples. We can store annotations or metadata on the column level. We can also store metadata on the data set level, and it can get quite rich. We can have full README, so you can really document everything that's happening within this data set. And even though the table is a good mental model, if you look at it technically, we use something called ZAR. I don't want to get into the technical details here, but I'm really proud of what we've come up with, because it turns out that this is like a data format that is applicable to any modality we've encountered so far, at least, and we're excited to be proven wrong and improve our system if there's something we can't quite do. But like from phenomics all the way to like just small molecules, like we can store all of it. And we can also work with very small data sets, so a couple hundred of kilobytes, to very large data sets. The one we've been using internally as our stress test is RX3. This is microscopy imaging, this phenomics data set that's 83 terabytes, and we can scale to that size. And why is that exciting is because you can do all of this with like one unified interface. So you don't have to worry about the data format. You don't have to worry about the size of the data set. You can load the data set with the same lines of code. And I really think that's important. Again, it feeds into the argument that things need to be simple, right? Like if you're a machine learning scientist, you're looking to apply your skills. If you have to juggle all these different file formats, file parsers, chances are you're gonna either not understand or introduce little bugs. So like we want to take all that friction away and make it as easy as possible to work with the data. As I was saying before, we really want this to be a community-driven platform. So anyone can upload a data set. If you create an account right now, you can upload a data set in 5 minutes from now. That does create a bit of a tension, because how do we then distinguish high-quality data sets from low-quality ones? If anyone can upload it, how do we make sure that those, like, how do we distinguish signal from noise? This is, again, like this is like an iterative thing. I don't think we'll get it right on the first try, but what we came up with is these 3 sort of high-level criteria that make for a good data set to be used for machine learning. And then we implemented a review process that lives on GitHub. It's open to all. Everyone can go and chime in. And when a request comes in, we'll either review it ourselves or we'll find an appropriate expert that can actually speak to the quality of the data. And again, it's open. So if you don't agree with the decision, you can find the GitHub and chime in. And so this is like a very first hint of this, but like what's then the value of the platform? Well, for example, we can make sure that people only find certified datasets by default, right? This is one of the things we can do because we own the implementation of the platform. So if you're this average machine learning scientist who's just curious about the space, comes to the website, they will only see certified datasets by default and have to explicitly toggle to find datasets that are not of high quality. So this is like, it's very subtle, but it's one of the ways in which we are trying to drive people towards more impactful science. Okay, so we have datasets. Then we have a way to specify a task. We have a way to specify a split, and we have a way to specify metrics. And what that makes for us is a benchmark. And that leads to the main API of the Polaris platform. So you can see with one line of code, we can load the benchmark. And again, this works independent of whatever modality or the size of the data set you're working with. With one line of code, you get the train and test split. And these are like the actual exact same train and test splits, like 100% the same. You can't, like, there's not one index of difference. Then with one line of code, you can evaluate your predictions. And with one line of code, you can upload those predictions to the hub to share them with everyone else. And so what we're trying to do as a first step is to really standardize and become like operate as a source of truth for benchmarks within the space, where people can be confident that if they generate their results like now or a couple months from now, they're the exact same results. We want to lower the surface area where possible mistakes can creep in, no slightly different implementations of metrics. Like we want to really like standardize as much as possible. The code I'm showing, it's a Python client, it's open source. We have a dedicated team of engineers that are maintaining it. If you have any issues, please let us know, we'll fix them. And yeah. So what does that look like? Okay, so we have UIs for all these artifacts, right? So this is what the benchmarking page looks like. You can see there's a leaderboard with the results. It's not perfect, we're working on it, but like it's a first iteration. Okay, so we also have competitions, and we actually just completed the competition together with ASAP Discovery and OpenAtMe, and there's a talk about that later today if you're curious about more in-detailed results and outcomes. But for the sake of this talk, like what's the difference between a benchmark and a competition for us? So the main difference is when you get feedback. So for a competition, we limit when you get feedback, and that can take different forms. It can be only after the competition ends, or maybe only for a certain number of submissions, or at certain points in time. Whereas with a benchmark, the feedback you get is immediate. Now that has consequences. That means that for the competition, we want the data set to be blind. People shouldn't be able to just get that feedback in another way. And also, it means that for competitions, the evaluation needs to happen on our system. It can't happen on the client system, because then we would have to share the data set with them. That speaks to something else. So what are you actually submitting? What is it that we're evaluating with our system? So we've started out as something like very simple. So with benchmarks, you do the evaluation locally using our library, but you do it locally, and you just submit the result. That's a very low fidelity result, but it's, again, like it's focused on simplicity. We wanted something that's easy, and we'll build out more complexity along the way. So what we're working on, or what we've already worked on with competitions, is now you can just submit your predictions. So that's slightly better. You submit your predictions, we'll evaluate them, and we'll produce the results. And what we're working on actively as well is model submissions. So it's even slightly higher fidelity, where like, you submit your models, we run inference based on the predictions, we compute the results. And what we ultimately want to get to, but that's a complex question to do in an easy way, is code submission. You submit your code, we train your model. Once we have your model, we run inference, then we compute the results. Okay, so everything you've heard up until now, think of it as like a first proof point. We believe that we have some first ways in which we can nudge the community towards like more rigorous evaluation, but we're not quite happy with it yet. So for example, that preprint that I showed on like method comparison guidelines, like one of the things we recommend is to do cross-validation and to use statistical testing to compare results. But if you have a leaderboard, just like simply ranking your methods, that's sort of like, that's the opposite of what we're recommending, right? But again, now that because we own the implementation of this whole platform, what we're actively working on is an evaluation dashboard that no longer just presents a leaderboard where everything is ranked A to, well, 1 to N, but that lets you more interactively analyze these results, run statistical tests, figure out, look at the plots of the result. Like that's really what we want to be getting to, and we think that's important to really drive the point home. Okay, so all of what I've just said, you don't have to take my word for it. You can try it yourself. You can go to polarishub.io or scan the QR code, and what you'll be able to do is just create an account. You don't have to create an account, but you can explore the platform. If you at some point you want to participate in a benchmark, or you want to upload your data set, or you want to upload a benchmark, you want to run a competition, all of those things are possible. Feel free to reach out to us. Okay, so the last one is community. And this is really about how can we incentivize or popularize these best practices. And again, I was saying, like, I'm not sure if community is the right label here. Feels a little vague, maybe not entirely clear what's meant by it. So let me exemplify some of the ways in which we've been doing this. So one of the examples that came up earlier is already something we discussed, right? Like we don't want this to be a authoritative, like we don't want this to be like just us doing all the work and sort of leaving at that. Like this needs to be open science, it needs to be open. People need to be able to give feedback and need to be able to disagree with us and need to like join the effort and become part of this initiative. Another way is that we think it's important to provide both substance and form, right? So like, I think as scientists, one of the ways we like to publish our science is through publications, naturally. But I think we need to acknowledge that for a lot of people, that's not the only form through which they consume content, or perhaps not even the best form. So we're experimenting with different ways in making science more accessible, whether that's a webinar that we host to sort of talk through the paper and give people the opportunity to ask questions, or whether it's through blog posts, or maybe even like videos or animated videos, or I don't know, like everything is possible. We're just experimenting with ways of making the science more accessible. And this is a bit forward-looking, but I do think conferences and journals have a lot of power in our space. And I think if we can get some of these journals to adopt best practices, and we're actually quite far advanced in some discussions on this, in their review process, that can be a major driver of people actually changing the way they do research and changing their evaluation logic. If we can't convince existing journals or existing conferences to do so, maybe it makes sense to start our own conference, or maybe it makes sense to start our own journal really focused on benchmarking. I was at a conference 2 weeks ago in Switzerland, and it was more bioinformatics and machine learning and more like fundamental biology than drug discovery, but they actually had a conference on benchmarking, and I thought it was awesome. It was so cool to see, like, to hear how people have been thinking about this and to see how complex it is. And like, I was just wondering, like, why have we never done that before? And we are seeing some workshops pop up at conferences, but I think this is something we could use more of. So with that, thank you all for your attention. If you want to get involved or learn more, you can write us an email or find us on any of these platforms. Thank you.